Assalamu alaikum everyone. So welcome back to my channel. Today we are going to discuss very important topic in genetics, that is the SNPs, that is the single nucleotide polymorphism. Single nucleotide polymorphism can also be pronounced as SNPs. So what is SNPs? A single nucleotide polymorphism is a variation at a single position in a DNA sequence among individuals. What does that mean is these SNPs are the small, these are the tiny changes in the DNA, but they can have big impacts on our health, on our traits, and even these tiny changes can make us unique. Now, let's see this particular individual's sequence of a DNA. This is the, let's say this is individual 1, this is individual 2, and this is individual 3. Now, in when we talk about SNP, what is SNP? This particular individual having a this sequence of the DNA. This particular individual containing this sequence at a specific chromosome at a specific gene position. Now, if we see in the individual 1, this individual carry A at this position, and this individual it carries G at this position, and the third individual is carrying the T nucleotide at the same position. What does that mean? These three individuals, when we talk about the same chromosomes of these individuals at the same chromosomes at the same gene locus at the same gene position, these three individuals basically carry the different nucleotides. For example, this is carrying the A nucleotide, here it is carrying the G nucleotide, and here it is carrying the T nucleotides. So these SNPs, these are the most common types of the genetic variation among the people. These occur, why these are called SNPs? Because these are the single nucleotide polymorphism. They occur at a single nucleotide, at a single position. They occur when a single nucleotide that is replaced by another nucleotide at a specific position in a genome. For example, let's say if most of the people have this particular sequence of the nucleotide, when there is adenine, then there is thymine, then cytosine, and guanine. Now, if most of the people having this particular sequence at a, let's say, at specific position on a at a specific locus on a chromosome, but you have this A, then this T, but instead of C you have this G, and next there is also G. Now that's the SNP. So here you can see at this particular position, this many of the individuals are having this C at this position, but you have this G. Now, this single nucleotide change at this specific locus, at this specific sequence, is basically making you the unique to unique. So these SNPs basically occurs approximately once in every 300 nucleotides. Means you can say that human genome roughly contains 10 million SNPs. So what does that mean is SNP is basically a change in the single nucleotide at a specific position, because it's a kind of a polymorphism. Poly means, when we talk about poly, what does that mean is poly means many, and morphism means forms. So these many forms of the same nucleotide. So morph, when we talk about morphism, it means it has many different forms. And when more than 1% of the population, let me write here, when more than 1% of the population is containing that SNP, that particular changed nucleotide, then it will be classified as a SNP. For example, here you can see in these three individuals, at this particular position, this there is A. At this particular position, there is G. Because we are talking about the same position, but these are the three different nucleotides at the same position. That's why it is classified as SNP. Now the question is, where do these SNPs occur? Now these SNPs can be part of the coding region, these can be part of a non-coding region. Coding region are those regions because these can be the gene. They can form a final product, that is the protein. And the non-coding regions, basically they do not form any kind of protein. These non-coding regions, they have just you can say the regulatory effects on that particular proteins. Now in coding region, these SNPs, they can change amino acid. For if they change amino acid, then there is altering of the protein function. But in the non-coding regions, they can affect only their regulation, they can affect their splicing. Now these SNPs can be of many types. You can say these SNPs can be present in the coding regions, as well as these can be present in the non-coding regions, and these are also present in the intervening regions between the genes. These are known as the intervening SNPs. Now when we talk about the um, synonymous SNPs. What are the synonymous strips? These are the changes in the DNA, but not the protein. There is a specific change at the specific position in particular DNA, but this DNA will not alter the any protein. Whereas the non-synonymous SNPs are those that can result in different amino acids. What does that mean is they can alter the proteins. Now, these non-synonymous SNPs can be further classified into the missense SNPs and the nonsense SNPs. So when we talk about the missense SNPs, now it can be result in a different amino acid. Now, this missense SNP, it can be result in the different amino acid. But this nonsense SNP, it will result in the stop codon. It will result in the stop codon. Now, what does that mean is this nonsense SNP that is occurring in the coding sequence. This can be introduce, you can say, a premature stop codon. Now, why should we care about SNP? These SNPs can be used in the genetic testing. Let me write here. These SNPs can be used in the genetic testing. For example, they can use in the ancestry test. These SNPs can be used in the pharmacogenomics. This can be used in the pharmacogenetics or pharmacogenomics, because these help us to predict they help us in predicting how a person will respond to a particular drug. And after that, these SNPs can be used in the research, in the disease research. For example, these SNPs can be used in identifying the mutations that are linked with the cancer, diabetes, etc. Now, this is all about the SNPs. And yes, very important thing, these SNPs, these can be different. These are basically different from the mutations. These SNPs are those which if more than 1% of the population is having the same change at the same gene locus. But in mutation, these mutations can be in the less than 1% of the population. These mutations can be rarely occur in a particular population. So that's why this is the main difference of the SNP. And the SNP, it is not necessarily that the SNP always cause any particular disease. So this is all about the SNP. Now, if you found this helpful, then please don't forget to like, comment, and subscribe. And thank you very much.